The pupillary light reflex controls the size of the pupil in response to light. When light is shined into one eye, both pupils constrict. This reaction helps protect the retina from excessive light and optimizes visual acuity in varying levels of illumination. The reflex begins when light enters the eye and causes stimulation of the optic nerve, which carries information about light from the retina. The optic nerve becomes the optic tract, and a subset of optic tract neurons project to the pretectal area in the brainstem, where they synapse on neurons that extend to nuclei called the Edinger-Westphal nuclei. There is an Edinger-Westphal nucleus on each side of the brainstem, and these nuclei contain neurons that project through the oculomotor nerve to a structure called the ciliary ganglion. From there, the short ciliary nerves extend to the pupillary sphincter to constrict the pupil. Because the signal from the pretectal area is sent to both Edinger-Westphal nuclei, shining light into one eye results in pupillary constriction on both sides. The response in the stimulated eye is called the direct pupillary light reflex, and the response in the opposite eye is called the consensual pupillary light reflex. Clinically, the pupillary light reflex is used to assess the function of the optic and oculomotor nerves and related structures. For example, if light is shined into the right eye and neither pupil constricts, it suggests a deficit in sensory input and may indicate damage to the right optic nerve. But if light shined into the right eye causes only the left pupil to constrict, this indicates the sensory signal was received since the left pupil constricted, but it may suggest a problem with the right oculomotor nerve, since the motor signal does not seem to be reaching the pupillary sphincter.